From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 5, recorded on January 26, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Greetings, Vincent. Great to be here again for a lovely day for a midwinter podcast.
Yeah, we've had some exciting times here on the East Coast.
Sounds like it. Glad to hear from you at all today, given the
really
blizzard-related issues you guys have been dealing with.
Well, I'll fill you in as soon as we bring on our two guests. Over on TWiEVO, sometimes we start chatting before we introduce everybody.
Yeah, let's get up to speed here. I'm excited for our
They're very
crew.
patient. We have a great set of guests today. One of them happens to be sitting right here in the TWiEVO studio with me. He's someone who's been on TWiEVO before. He's a professor at the Albert Einstein College of Medicine, Kartik Chandran. Welcome, Kartik.
Hi, thanks for having me on the podcast.
You know, Kartik, there's a Kartik Chandran here at Columbia.
Yeah, it's unfortunate. And he's a MacArthur Genius Fellow.
You know, when I go to send you an email, I just type KA, and then his email comes up, and I send him emails, and he's very patient. He says, I get these all the time.
I was hoping that I'd get his check by mistake or something. But
no, his MacArthur check, right?
Yeah, that's right.
Well, maybe next year they'll add a second Kartik Chandran to their roster.
Actually, then there's another guy. There's an astronomer or something. Because I Googled you, and I got a bunch of Kartik Chandran. It's like Vincent Racaniello. It's a common name. So anyway, Kartik was on TWiEVO number 314, which wasn't so long ago, right?
Yeah.
Our other guest is out in Boulder, Colorado. She is at the University of Colorado. She's a professor there. She's also in the BioFrontiers Institute, and she has also been on TWiEVO. Sarah Sawyer, welcome.
Hi, great to be here.
You were on TWiEVO 193.
Filmed live at ASV.
In Madison, yeah.
Yeah, that's right. That was good fun.
So I was out visiting Carolyn Coyne last week, and we were reminiscing about that TWiEVO, and she said you went first and started talking about how you used to work on an oil rig, and she said, what am I going to do to follow this up?
Yeah, that's funny that you should say that, because I really don't like to think about that TWiEVO very much, because Carolyn really showed me up, I felt.
No, I don't think so at all. I think she thinks you showed her up, so you're both even, no problem.
I remember that episode. I was at that meeting also. That was really fun. And actually, that's where Kartik and I first met, by the introduction of Sarah.
I think
the night before we filmed that, actually, yeah.
There was a lot of drinking involved, I seem to recall.
Yeah, a few details are a bit fuzzy. I'll confess to that.
Yeah, that's right.
Lots of connections in science, and now to all the other connections that we have, and we're not many degrees of separation apart. Now we add podcasting. It's really funny.
Yeah, that's right.
Well, I'm really glad, and so is Nels, that you guys could both make it together at the same time. It's always hard to schedule people, so this is great. And we're going to talk about a paper that you guys have co-authored that just came out. But before we do that, Nels, you want to know what's been going on here in New York City?
Yeah, I've heard some rumors that you guys have been sort of visited by winter.
Yeah, we got a big snowstorm, Snowzilla. 2016 started, and Kartik, you were here in the city too, so you know all about it, right? It started Friday night, right?
Yeah.
So I wasn't here. I was actually in Pittsburgh. I went to Pittsburgh on Thursday morning. I got there, and Thursday night my flight back was canceled. So that was going to be Saturday morning. And so I spent Thursday and Friday there, and everyone's telling me, go home, but I didn't want to disrupt their schedule. And I booked the train for Saturday morning. I'd actually booked a car, but I decided not to drive.
Good idea.
I got on the train at 7:30 in the morning. Got to New York City at midnight.
Wow.
Train broke down a couple times along the way. We're just sitting there in the middle of nowhere with no heat.
Man.
They had to change engines. Finally got to Philly. They said, we're going to change the engine again. And that didn't work. We got on another train. And then I get to New York. I can't get back. I live in New Jersey. I couldn't get there because all the trains were shut down. There's no driving. So I stayed with a friend here in the city, got home Sunday evening.
Oh, boy.
Wow.
And then Monday morning I went to the airport and finally got my car and brought a shovel with me and dug it out.
Oh, wow. It's like planes, trains, and automobiles.
You weren't impacted at all, Kartik, right?
I just had to shovel my car out, but
Yeah.
not too bad.
But we have big piles of snow here. We got 30 inches or something.
Yep.
And the problem here in New York City is they just push it around. They clear the streets, but if you're a pedestrian and you want to cross the street, you have to find a little gap in the snow.
Yeah, pretty much.
And then when you walk on the street, you usually sink in snow a few inches. And
There's that layer of slush that is sort of invisible that you can step right into.
Oh, yeah.
Do you have snow out there in Salt Lake?
We do. And so, excuse me, I have a slightly contrasting winter blizzard-related story here. So about the time when you guys were getting hit, we had a different storm, not Snowzilla by any means, but around, I think, Friday night or Saturday, it started raining in the valley, which is sort of this perfect situation for Salt Lake where it rains in the valley, but then there's this great dry, powdery snow falling in the mountains. And so Sunday, I ended up heading up to the Alta ski resort and took advantage of some of this. I don't have any sort of harrowing stories like you do, Vincent, of trying to to navigate across state. But what did happen is I got into this really nice gully and thought, oh, there's some great powder here. I'm going to go up onto the edge, and not noticing a few rocks that were a foot or two under the surface of the snow, and I got de-skied. Both skis stopped, and I kept tumbling. So that was the drama here, but sort of a much more kind of celebratory version of hanging out with snow versus some of the real headaches that you guys have been up against.
You got any snow out in Boulder, Sarah?
Not as much as I would like. I spent the last 8 years in Texas, so I was ready for some snow. And, it's been incredibly dry the whole, 6 or 8 months I've been here. We just have a little bit of snow on the ground. But I'll tell you, it's kind of fun. I live really close to the university, and when it does snow here, all the students at the University of Colorado ski and snowboard down the street in front of my house. So that's pretty fun. And some of the crazier ones even hook up their snowboards to their buddy's truck and, let them themselves be pulled around the neighborhood. So anyway, it's keeping me young.
So the last time, when you were on TWiEVO last, you were at the University of Texas, Austin, right?
Yeah, that's right.
And so recently you moved to Boulder.
Yeah.
And what is the BioFrontiers Institute?
Yeah, so we came out here to work in this institute that was started by Tom Check after he left the directorship of HHMI a handful of years ago. He started BioFrontiers, and basically his mission statement is pretty simple. He says that if you look at the job ads for assistant professors, a lot of what molecular biology departments are looking for are people trained in the intersection of experimental biology with either computer science or physics or engineering or any sort of these, hard science disciplines. And so the mission of this institute is to train a whole group of graduate students to come out with those skills. And Tom told me when he was recruiting me that the only way he knew how to do that was to hire an institute full of people that were trained at that intersection. So he's kind of trying to build this really fun interdisciplinary place, and it's been a blast being here, actually. Yeah.
So you don't regret leaving Texas, huh?
In many ways I do. I actually really loved the city of Austin, and I had some great colleagues there, but this is a new environment. That's always a lot of fun. And in a way, I brought half of Texas with me. My whole team came with me here, and a nanny came with me. Even our grants administrator came out here with us. That was in the department there. So it kind of feels like I never left. I've got a lot of good people around me still, so that's the most important thing.
Where are you from originally? Remind me.
Yeah, I'm from Kansas City, Kansas.
Okay. You went to Cornell, right?
I went to Cornell for my PhD, but I went to my local state school for my undergrad. I went to the University of Kansas in Lawrence.
Okay.
So, and Vincent, I can vouch for the BioFrontiers Institute being a pretty interesting and spicy place to do science. Yeah, Sarah invited me out a few months ago, and my goodness, it was really full of energy. It's really vibrant. And how Sarah's describing it, sort of, bringing people together who've trained across disciplines. And I think Sarah fits really nicely into that category, combining evolutionary approaches to experimental approaches in virology in particular.
Now, Sarah, you were a postdoc with Harmeet Malik, right?
Yeah,
You were his first, correct?
I was. I was the first person to join the lab, and then I tried my hardest to recruit Nels so I could have some company there. Actually, there were a few people in between, but the other part of that was true.
So, Nels, you went as a postdoc as well, correct?
I did, and I remember very well. So I was kind of finishing up my time in grad school, and it was, grad school I think is pretty tough on a person. You have some ups and downs, and so in finishing it up, trying to get your thesis together, thinking about all of the next steps, there's a lot going on. And so I came to that interview, and Harmeet's lab was very new at the time. He had just opened his lab after being in Steve Henikoff's group just next door, actually, at the Fred Hutch for his postdoc. And it was really a breath of fresh air. And Sara in particular, just seeing how she had already transitioned from grad school to postdoc and was just having a blast and doing really fun, interesting things. And it was just rejuvenating. And I remember leaving that interview thinking, wow, if I could get a job here, this would be sort of a really fun step forward. And that turned out to be right when we overlapped for a few years and had a really great time.
Nice.
Since we're all throwing praises in Harmeet's direction, I'll just say I remember I was actually a graduate student at Cornell when he interviewed for a job as an assistant professor there, and I didn't want to go to that seminar because I was just days from my dissertation defense and I was working, and my advisor made me go, and I sat next to her at the seminar, and as soon as the seminar was over, I turned to her and I was like, I'm working for that guy no matter where he goes. And right after that, I got to have the student lunch that they always do with the candidates. And I realize now this must have been a really kind of intense from Harmeet's perspective. But I remember at that lunch, I said to him, wherever you decide to go, I want to come be a postdoc with you. He's probably sitting there thinking, I don't even have a job yet. What are you talking about?
I think you made the right decision.
Yeah, it was a great time. Man, he's a great person. Yeah.
So Kartik, let's just review your history a bit. Where did you were born in India, I think, right?
Yeah, I grew up in Bangalore.
And how old were you when you came here?
I was 17. I decided that, in 7th grade that I wanted to be a biochemist.
Mm-hmm.
And I used to annoy all my biology teachers by asking them to explain the chemical basis of heredity and genetics, and they just tell me to sit down. So I just kind of applied to one school, early decision, which is Lafayette College in Pennsylvania, and was completely shocked to get in and get a scholarship. So came out here when I was 17, did a bachelor's in biochemistry at Lafayette, which is in eastern Pennsylvania, and then went to UW-Madison for a PhD, where I worked with Max Neibert, who is a double-strand RNA virologist. You trained with Bernie Fields.
Right.
And moved out with him to Boston, Harvard, for a postdoc. And, do you want me to keep going?
Then after your postdoc, you moved here.
Yep, I did a postdoc with Jim Cunningham, who was a retrovirus guy at the Brigham, and then came to Einstein after that.
So it was with Cunningham that you started to work on Ebola, right?
Yeah, so that's where the Ebola, we started working on Ebola. I sort of joined his lab to work on retroviruses, but about few months in, I decided I didn't like retroviruses that much and wanted to do something else. So, and we're like, hey, Ebola, it's pretty interesting. We don't know that much about it. Let's start working on it.
It's interesting. There are a lot of small, good colleges in Pennsylvania besides Lafayette, and one of them is Gettysburg.
That's right.
And that's actually where Mike Bishop went to college.
Oh, yeah.
They interviewed him a year or so ago and was talking about it. And I know because a lot of kids in my kids' high school, they all want to go local, and a lot of these small Pennsylvania colleges. Lehigh is another one.
Right.
Franklin and Marshall. And there are many, many more. I don't know why there's so many. Well, I went past all of them on the train the other day.
You just made the tour.
Exactly. Well, Nels, I put a couple of photos of New York snow in the show notes.
Yeah, I saw that. And it kind of puts a visual impact there on the idea that in New York, when you get that much snow, there's not much place to put it. And so it starts to pile up. Yeah, wow.
So the reason we're here today is because Kartik and Sara have co-authored a paper which was published very recently in eLife. And Kartik, you sent me the manuscript a while ago, and as soon as I looked at it, I said, "This is going to be a podcast."
Cool.
And I was targeting it for TWiEVO, but then, it's got a lot of things that Nels is interested in. So I said, "Why don't we do this on TWiEVO?" And before I knew it, he had you guys both booked.
I couldn't resist. I mean, Sara has been telling me about this manuscript for a while as well, and so I was thrilled to see that it had come out, and excited to do this. And I think it is sort of a hybrid program in a sense. Maybe this is a new category of podcast, Vincent, called TWiEVO.
Yeah, you bet. So it's, as I said, it's an eLife, and it's called Filovirus Receptor NPC1 Contributes to Species-Specific Patterns of Ebolavirus Susceptibility in Bats. It's a really cool paper, which we'll talk about. A lot of authors. And I'm just curious how... So Kartik is the last author, and Sarah's the second to the last. How did you guys get together on this? Do you remember the moment?
So, I mean, we found that NPC1, Niemann-Pick-C1, which is this, cholesterol transporter that's present in, pretty much all animals, and is really an important human disease-associated gene, also has in moonlights as an Ebola receptor. And from that point, I mean, I had actually heard two different talks, one by Harmeet Malik and one by Sara at ASV, I think, especially Sarah's work on virus receptor coevolution with arenaviruses. And, so it really jumped out at me that was something that we wanted to pursue with NPC1 to try and understand whether the virus, like the relationship between filoviruses like Ebola and NPC1 are somehow driving evolution. And so I actually reached out to Harmeet first and to talk to him about it. And Harmeet immediately said to me, you should work with Sara on this, because Sara is really into this kind of question with looking at virus receptor interactions. So I reached out to Sara, and she was very interested, and we started working on this together.
Yeah, and I'll just interject and say, working with Kartik was a no-brainer. We actually didn't know each other at that point, although we know each other very well now. How many hundreds of phone calls have gone into this one, Kartik?
Yeah, when you try to publish a paper and it takes 6 journals to do it, you get to know each other very well.
Yeah. That's right. That's another story. I didn't know we were going to admit that on air.
Isn't that the average for everybody?
Probably.
Yeah.
There's no secrets on TWiEVO.
That's right.
This paper has a number of authors, and it's like a who's who. There's so many people I know here. Now, I know Rohit, who's your postdoc, right?
Yep.
Kartik, he's a big TWiV fan, and he's always writing and correcting me.
Oh, yeah.
Robert Gifford is in Glasgow, right?
Yep.
All right, I've met him, and we talked about one of his papers on TWiV. I know Thin Brummelkamp, of course, from the haploid cell fame. Christian Drosten, who had at ASV last year. Linfa Wang, the original Batman. Jens Kuhn. Now, how did Jens get on this? Maybe you worked in his BSL-4. That's why. We'll see. Sara and Kartik. So it's quite. Do you know anyone else on this, Nels?
Yeah. Well, who's your third senior author?
So that's John Dye. Mm-hmm. So John is our go-to guy for all things BSL-4 related.
Yeah.
We got together a couple of years ago to work on the NPC1 stuff, and we've become really good friends. And now we're thick as thieves on every project. And so there are some live virus experiments in here that John's lab did.
That he's at US AmRed, right?
That's right.
Okay, so that's where you did the BSL-4 experiments down there with his.
Yep.
And that's where Jens comes in as well, I guess.
Actually, Jens and I have been talking. Jens is kind of like a guru of filoviruses in that he's really understands the sweep of the field and the history, and is really interested in this idea of what kinds of animals could be hosts for viruses like Ebola, and also the evolutionary piece. And I think we've really had many conversations about this over the years that we've known each other. And he's also incredibly well connected and was able to get us a lot of the cell lines and things from bats that are pretty esoteric and all over the world and hard to corral. And so in part I think that also reflects the author list, in that several folks on there gave us these unpublished reagents that we were able to use for this study.
So Go ahead, Sarah.
So I was just going to say, you asked about how this project began, and there's kind of an interesting story there. We got these 4 cell lines from 4 bats that are in the phyloendemic zone of Africa. That's how the paper starts, is looking at which one of those cell lines can support Ebola infection. And pretty quickly we figured out that Niemann-Pick was sort of that's the receptor that Ebola uses to get into cells that Kartik and others discovered some years ago. We figured out that was the critical difference that made some of these cell lines susceptible and not others. And so we started focusing on that. And one of the first things we needed to do was sequence that gene from those 4 species. And that sounds simple. And in fact, my lab does, sequencing of sort of exotic species that don't have genome projects all the time. We sequence genes from all kinds of animals. And so we know how to make degenerate primers and go after things that are a little bit more divergent. But I gave this project to a brand new graduate student, Mariska Kaczmarek, who's now one of the first authors on this paper. And it actually took her a year and a half to sequence these 4 bat genes, because the 2 bat genome projects that were available at the time, we couldn't even find this gene. The closest mammal that had an annotated Niemann-Pick gene was the horse, and we couldn't even really get it to align to anything very well in these bat genomes. So it's a giant gene, and we had to piece it out little by little. So, that was probably 3 or 4 years ago that project started, and just getting it off the ground. When you're talking about species like this that are not laboratory model systems, in fact, not even related to anything that's a laboratory model system, everything comes at a little higher capital, actually.
So this actually started 3 or 4 years ago.
Yeah.
Wow.
Yeah, I think we might have started in 2012, Sarah.
Right. And in fact, the bat genome projects kind of lapped us during the progress of the project, because it would be quite a bit easier now, but it wasn't easy back then. Yeah.
So these bat cell lines, which you mentioned, Sarah, you can work with those obviously in BSL-2 conditions, right?
These are actually immortalized lines at this point, right, Kartik?
Yep, these are immortalized.
Yeah, so we do.
But we also got some primaries as well, and we're trying to get our hands on a lot more. It's pretty hard to make these cell lines from samples that you get out in the wild, unless you're working with the zoo or something. Yeah. So there's all kinds of logistical issues, and I think in the future, to extend these kind of projects, we would really like to come up with ways to do that better.
Well, actually, we had a segue in the middle of this project where Kartik and I put down 20 grand on the table to get a group to trap a bunch of bats for us in Africa. Wow. We had a big trapping project where a group trapped something like 35 or 40 different species of bats for us. But those samples had to be processed in Africa, because we couldn't get the paperwork approval to import those samples to the United States as tissues. So we had the nucleic acids extracted out of those in a lab in Africa, and the quality from that particular lab just on that particular day wasn't good, and sort of all the samples were ruined. It was really tragic, actually, but this is the kind of difficulty you run into on projects like this.
We also, went to my student Esther, who is one of the first authors, and I went to Uganda with John Dye, because they have a project they're following Ebola survivors, and so we kind of piggybacked on that trip, went along, and we're actually able to hook up with a bat zoologist there. And so we've started a relationship that I'm hoping is going to allow us to obtain, many different kinds of samples and cell lines from different kinds of bats, and probably other mammals as well. I think with Ebola, people fixate on bats, but I think it's probably a lot more complicated than that.
Yeah, I wanted to ask you about that. You discussed in the paper the evidence that bats are a reservoir, and it's pretty slim. It's a little bit of serology and fragments of nucleic acid, right?
I mean, I think the data are really the best for Marburg virus, which people always think of as kind of, like Ebola light or something. But I mean, this virus is quite divergent from Ebola. And so work by John Towner at CDC and others like Janusz Paweska in Johannesburg has really led to this idea that this particular kind of bat called the Egyptian rosette or Egyptian flying fox could be a reservoir for Marburg virus. And there's certainly a lot of evidence now that, this particular kind of bat, which is a cave roosting bat, can harbor Marburg virus. But there are still many unanswered questions even in that case, because for reasons that we don't really understand. I mean, there's a recent study from Paveshka showing that if you take a colony of these bats that now people are working with them in labs, if you inject Ebola into one of these, actually Marburg into one of these bats, and they all like to stick together very close, so there's a really good chance for horizontal transmission of the virus from one bat to the other, but they don't really see that. So it's a bit hard to understand how Marburg is spreading from bat to bat within these colonies. So I think, at this point we really don't understand very well the ecology of this virus.
So
out there.
when an outbreak begins, isn't it often bush contact with bush meat, not just bats, but maybe monkey tissues and so forth?
So actually, it's quite surprising that, even though people say that, there's actually, I can only think of a couple of instances where that's been really clearly shown to be from bush meat. I mean, it is speculated often, but, the evidence isn't necessarily
Has anyone
that strong.
looked for viral sequences in bush? I know it's hard to do, but they've done it in bats, right?
Yeah, I mean, every time there's one of these outbreaks, folks from the CDC and other places, fan out around the outbreak sort of area to sample wildlife and things like that. But, at least with Ebola, a lot of times they come up empty-handed.
Right.
Yeah, and just how you're describing, and we're talking about all of the authors and the contributions of going and sampling in Africa, the diverse species, sort of the difficulty of even detecting the sequences. That was one of the things I really enjoyed about the paper, is just how this reflects almost this collage of different scientific approaches and expertise with the BSL-4 scenarios that we'll be diving into in more detail. It's really fun to see that. And then how kind of, I think you also see this complexity and sort of the evolutionary stories echoing on as well.
Yeah.
I think we have to, sorry, go ahead.
Yeah, I'm sorry. I just wanted to say, this was my first research project on Ebola, and I was instantly captivated by this project. Why are you laughing at that?
It's funny.
Okay. Well, anyway, there are two sort of great enigmas in this field that I find to be very interesting, and maybe we can use it to frame this conversation a little bit. But, the interesting thing about Ebola, as Kartik has educated me over these years, is that Ebola will infect basically every mammalian cell line in the lab, yet we find it nowhere in nature. So, we've never identified this virus from anything other than what we believe to be a terminal host, like an ape or another African primate or humans, which we all believe, are not the long-term reservoirs, although maybe that's not true for all primates, but so far that's not been established as a reservoir. So, what's up with that? Why are cells so universally permissive, but then we find this virus nowhere in nature after all these years of looking? And then, the other really interesting thing is that in every other case of a major human pandemic or epidemic virus, we usually have a fair amount of sequences from the virus in its reservoir species, where we can sort of with HIV, we have all kinds of SIV sequences from different primates in Africa, and we know exactly which primate, was what contributed the virus that was the progenitor of HIV-1 and HIV-2, and we have all this natural history on the virus going back into its reservoir, and that's just a giant black box for this virus. Right. Nobody knows where this thing is, and so when people try to do these phylogenetic studies on human sequences in the current outbreak or past outbreaks, there's an analogy that I like to use. When I lived in Seattle, you would go skiing up in the mountains and there would be these huge sequoias, but there would be so much snow on those mountains that all you would see would be the very tip or maybe a couple little tips of the tree. And it would look like there were 3 little bushes there, but really it's a giant sequoia tree, right? That you're standing on top of. That's the situation with Ebola. These outbreaks are little tiny bushes that we see that are branching off of an enormous sequoia tree that's underground and invisible to us, and we know nothing about it. And the Ebola outbreak sequences that we have are, in the grand scheme of things, virtually identical to one another, because these outbreaks don't last for very long. Nothing much happens to the evolution of these viruses. We really need to learn more about that underlying tree, and we just know nothing.
I mean, it's clear the virus is evolving in its natural reservoirs because as you kind of look at outbreaks over time, you can see the virus changing. And one of the surprising things about the 2014 outbreak in the DRC, Democratic Republic of Congo, which kind of got lost in the background of the West African outbreak, was that the virus that seems to have caused that outbreak is a throwback, genetically speaking. It looks a lot more like the original 1976 virus than the one that caused the West African outbreak, even though it's the same species of the virus belongs to the same species. So, there are really mysteries about what the virus is doing when no one's looking for it out there in nature, before it jumps into people.
So let's go through some of the data. Now, you were talking before, Kartik, about NPC1, this endosomal protein involved in cholesterol transport, which you and others have shown is an important receptor for fusion of the viral envelope. But at the cell surface, do we still not know much about what's going on there to bind virus?
So I think, there's actually now quite a bit of work showing that, from Wendy Maury, for example, Rob Davey, others, showing that, there are these TIM and TAM proteins, which are phosphatidylserine receptors that are often involved in engulfing apoptotic bodies, for example. So they actually detect phosphatidylserine, which is a lipid that's on membranes of little cellular blebs that are engulfed by cells. And it turns out that not just Ebola, but a number of other viruses have evolved to use that interaction. Essentially, it's like an eat me signal for the cell, and the virus is masquerading as a piece of junk that the cell is going to try and eat and break down and recycle. And so now this phosphatidylserine lipid is usually on the inner leaflet of the cellular membrane, but it turns out that when the virus replicates in the cell, probably because cellular stress, some of the phosphatidylserine flips to the outside. And when the virus buds out of the plasma membrane, it takes some of this lipid with it. So now you have these long Ebola noodles that are decorated with phosphatidylserine, and that allows the virus to stick to these receptors and then get engulfed. And so we think that that's actually a major way that these viruses get in. And, so that's sort of a more non-specific, I don't, when I say non-specific, I don't mean that it isn't a specific interaction. It's just not a specific interaction with the viral glycoprotein on the surface. So we think that once a virus has been engulfed and goes into an endosome, which is one of these intracellular compartments, sort of like the stomach of the cell, where all these things get pumped in that break down cargo and junk that's taken up by the cell, that the virus has to find Niemann-Pick C1 on the membrane of this endosome, and that's sort of like the GPS system for the virus to tell it when it needs to blow out of the endosome into the cytoplasm, where it needs to go.
Okay. Now, you start by looking at, you have these fibroblast cell lines from 4 different bats, right? Yeah. And the bats are from Africa. It's kind of different regions of Africa, a little bit of overlap in some case. Did you have what was the goal of that first experiment where you took actual filoviruses and infect these cell lines? What were you trying to find out?
So, Sarah said before that one of the sort of puzzling things about filoviruses is that they can infect really a broad range of mammalian cells in vitro. So, I guess we weren't really expecting very much when we did the experiment, because we figured it would just be like all the others. I mean, that all these different viruses would infect all 4 cell lines just fine. But immediately we saw this interesting result, an unexpected result, which is that one of the cell lines, which is from this bat called the African straw-colored fruit bat, or Eidolon helvum, was much less permissive to infection by Ebola virus, even though it could be infected by some of the other viruses like Sudan and Marburg. So that immediately sort of smelled like a host genetic difference that was really driving this difference in infectivity of the virus. And so we immediately kind of, went into hypothesis testing mode, and we're able to show that when we just take human NPC1 and express it in this eidolon helvam cell line, that we could restore that loss of infectivity. So that told us that it had to do with somehow the NPC1 in this particular kind of bat not being compatible with Ebola. But it was this very specific kind of incompatibility that depended on the virus and the species of the host. And that's the sort of thing that gets Sarah Sawyer very excited. So because
and so that's really an amazing, cool result. But and you say in the paper, and you kind of hinted at it already, that you sort of surmised the role of NPC1. Was that really the first target you tried, or did you consider other candidates at the outset of those experiments? I'm kind of curious how that
was the
happened.
First thing we tried.
Well, we had a major clue, right? Because the species difference was obvious with pseudotyped virus, so we knew that it was some sort of entry phenomenon, right?
Yes, we knew it was entry, but there are other host factors involved in entry, so it could have been something else. But you try different things, and sometimes things work, and sometimes they don't. In this case, we immediately found that NPC1 was the basis of the difference.
So we should explain for some people who don't know what a pseudotype is. Kartik, can you?
Right. So for if you want to work on Ebola virus, you have a couple of options. So you can work with the real Ebola virus, the wild type virus, but of course this is a highly lethal agent, and you have to do it under BSL-4 containment. And this is where my buddy John Dye comes in. But, we don't have that in New York City. Maybe we should, maybe we should...
It's not allowed.
Talk to the governor or something, I mean, why can Boston have it? But anyway, so we don't have that here. So, and, in any case, I don't want to lose any graduate students or anything. So what we do instead is we take advantage of the fact that the virus is actually a modular entity. And so there is a single protein that's called the spike glycoprotein that's sticking out of the membrane of the virus. And that glycoprotein is really a molecular machine that does everything from binding the virus to the outside of the cell to blowing a hole in the endosome and releasing the replicative core, the sort of the guts of the virus, into the cytoplasm, where it can reprogram the cell to churn out more viruses. So since that glycoprotein, and a lot of different viruses have this kind of glycoprotein, although that does the same job, although they all work by different mechanisms. And so what you're able to do, and what we're doing in this case, is taking the glycoprotein from Ebola and essentially putting it into a completely different virus called vesicular stomatitis virus, or VSV, that is a cattle virus, but you can grow it safely in the lab and work with it. And essentially, it's like a sheep in wolf's clothing. We have this innocuous virus, and we're using the Ebola glycoprotein to deliver its genome into the cell and kick off infection. And entry by this, and so this is called a pseudotype because the glycoprotein on the surface of the virus isn't really reflected in the genetic material that's inside the virus. So it's sort of a fake, so to speak. Nevertheless, because we are using the Ebola glycoprotein to get the virus into the cell, we can actually mechanistically dissect the Ebola glycoprotein in the context of this VSV particle. So that's a very powerful system for us to really do this kind of studies. The only limitation is that we can only study the entry step, which stops after the virus gets inside the cytoplasm. So if you want to look at other aspects of the viral life cycle, you've got to use other systems. But there are other such systems. So if you can sort of cobble together a bunch of different systems that allow you to study different parts of Ebola infection in a BSL-2 environment. And then once you've got interesting data, I pick up the phone and I call my buddy John, and he runs this in the BSL-4 suite. So that's a, somewhat cumbersome, but at the same time, we can do a lot more in BSL-2 than they can do in BSL-4. So it works out pretty well.
What was the first experiment you did with these 4 bat cell lines? Was it the pseudotype or the Ebola virus experiment?
Yeah, so even though we present it like we did the live virus experiment first, we did the pseudotype experiment first.
It makes sense, yeah. And those 2 pieces of evidence strongly suggest it's an entry. And then you did a kind of a complementation experiment to prove that, right?
Yeah. So we just put in human NPC1, and that complements the infection defect. So we know that it has to do with NPC1. Now the question is exactly why, and we look at that in the next experiment.
You also knocked it out in the bat cells as well, right?
Yeah, we did. So we used CRISPR. So I guess one of the questions that when we started doing this, we sort of were wondering if we were getting a little ahead of ourselves. I mean, how do we know that NPC1 is even a receptor in bats? Maybe it's something else. Maybe we're assuming too much.
Yeah.
So, my student Melinda, who is also first author on the paper, did a really nice CRISPR experiment where she used CRISPR-Cas9 engineering to knock out functional NPC1 in these bat cells. And she's able to show that these cells develop the same kind of cellular phenotype that happens in human cells, which is they accumulate cholesterol and lysosomes. And so that tells us that these cells do have a functional NPC1, that it is doing the same job, more or less. And when we look at viral infection, what we find is that even though these eidolon helvum cells are much less permissive to Ebolavirus infection, there is a little bit of infection there. And, we could talk about this a little bit more. This potentially could be important to think about. It's a non-zero infection in these particular cells. But when you knock NPC1 out with CRISPR, that residual infectivity completely goes away. And when you do the same experiment with Marburg virus, now you see that there is also no infection. So that allows us to conclude that NPC1 is indispensable for entry and infection by all these different filoviruses in bat cells. And so we're really starting to think now that NPC1 is a universal receptor for all of the viruses in this family. And the other sort of important conclusion is that in the eidolon helvum cells, there's a little bit of infection, which kind of gets us also thinking about this evolutionary arms race where the host is adapting to reduce the amount of infection that's happening by the virus. And so there's this back and forth.
Do you think that this residual infection is done by viral variants that have changed to adapt to the receptor in those cells?
Yeah, that's...
I know you did an experiment later to address that, but did you ever take those viruses that come out of the straw-colored bat cells and look at them?
So we've actually... That's a really good question. I mean, we've tried to enrich for those kinds of variants by just serially passaging the virus on those cells, and we've never managed to really find any. I'm not really sure why. Sometimes these selections work and sometimes they don't.
So it's just leakiness, right?
Yeah, I mean, most likely. Although it's clear that there are mutants that can do better on those cells.
Yeah. So you also did in this paper some binding experiments.
We did.
Which is not always easy to do.
Right. So it turns out, NPC1 is a giant protein. I think there's a figure in the paper later on that Sarah made where you can see that it's this really huge protein that dips in and out of the membrane. So it's a multi-membrane spanning protein, and sort of like the Loch Ness Monster or something. And it's got these
it does.
Pieces.
I can see the resemblance.
It's got these pieces that sort of stick into the endosomal lumen. And, a couple of years ago when we were trying to think about how Ebola is using NPC1 as a receptor, it seemed, really sensible to think that these domains that are sort of hanging in the lumen of the endosome like berries or something, at least in this two-dimensional picture, could be the pieces of NPC1 that the virus is interacting with. And so we followed up on that, and we were able to show that there's a single domain called domain C that sort of in the middle of the protein that actually is binding directly to the virus. And one of the nice things you can do is you can just genetically lop off just that little piece of NPC1 and express it and purify it as a soluble protein, and you can demonstrate that that soluble NPC1 domain C can bind to Ebola. Now, the other really interesting wrinkle to this interaction that we showed before is that it doesn't actually happen with the virus that's on the outside of the cell, because first of all, NPC1 isn't on the outside of the cell; it's only in these endosomes. But there's another sort of regulatory thing that needs to happen to the virus, which is that a big chunk of the spike glycoprotein needs to get cleaved off by enzymes, by proteases that are in the endosome, and removing this little, we call it a glycan cap. Erica Sapphire at Scripps solved the structure of this protein a few years ago, and we can infer that when you remove the glycan cap, you actually expose a receptor binding site for Ebola. And shameless plug, we have an MBio paper coming out with Erica in a few days that basically defines this receptor binding site. And so once a virus is in the endosome, the glycan cap comes off, the receptor binding site's exposed, and then it can bind to this domain C. And actually, there's another really important paper that just came out from George Gao's lab in China, but they actually solved the crystal structure of the domain C bound to this cleaved form of the Ebola spike-like protein. And, it's really nice, and it actually confirms a lot of our functional data. I sort of joked the other day to one of my students that, if we close our eyes, we could have painted the structure because we have so much functional data. But at the end of the day, it's amazing to have the picture to be able to stick all your Post-it notes on.
Yeah, that's really nice. And so, Kartik, you're describing this really cool way of sort of narrowing down through kind of the biochemistry and even the structural biology.
Right.
But you also alluded to this idea of these sort of molecular arms races or evolutionary arms races.
Yes.
And so maybe we could bring in Sara to sort of start to fill in that side of the story and how you go from sort of this domain level to the next step with this evolutionary perspective.
Yeah, right. So, we knew this gene was important. I'll call it, this gene, through the studies that Kartik just described, we narrowed down on the idea that this gene was an important determinant of species tropism, which means that not all bat Niemann-Picks are the same from the perspective of these viruses. So these viruses can enter cells through the Niemann-Pick of some species, but not others. Well, given that, you can pretty much guess that the protein sequences of Niemann-Picks from different bats aren't going to be completely identical, because if they were, they would be functioning the same. So at that point, we started by aligning these sequences from the different bats and trying to understand what could be contributing to this. And it's it's hard to, Kartik, how big is this protein? I've forgotten. It's huge.
Yeah, it's like 1,300 amino acids.
It's big.
Monster.
Yeah, right. So we took a look at it, and the idea here is that, any perfectly conserved positions, well, those are out. Those certainly aren't contributing to this different species. And I actually want to, point out, Kartik might not have thought to point this out. I just wanted to say it. This is actually the first, these Edelon helvum cells identified in this paper. This is the first mammalian cell line ever found to be resistant to Ebola infection. So it was pretty exciting to trace down the genetic and biochemical reasons for this. So that's why we tried so hard. So we aligned these sequences and we took a look. And so, you're basically going to have 3 types of evolutionary signatures in sequences like this. You're going to have perfect conservation of amino acids across all the bats looked at. We're definitely not interested in those sites. Then you're going to have other positions where mutations have happened that now differentiate the Niemann-Pick of species A from species B. And we can sort of subclassify those into to the final 2 groups. One group is just going to be mutations that really don't make any difference to the function of this protein, just random mutations that have occurred that didn't have strong selection for or against them. And then the third group would be mutations that have arisen over time that do impact the interaction of this protein with viruses. Now, why those mutations came to be selected is still a mystery. The fact that they do change interaction with these viruses puts forth a plausible hypothesis for why they could have been selected. I'll remind you that the paper shows that a single mutation at a single position in the Niemann-Pick protein of Edelon helvum protects this bat at the cell level from Ebola infection. That is definitely a property on which natural selection could operate. If a bat population is all of a sudden resistant to being decimated by filoviruses, that's a plausible idea for why that mutation exists in that bat. And so we looked, and there's a nice array of common statistical programs that people use to do these analysis, where they try to find positions and alignments that fall into that third category, positions where proteins from different species encode different amino acids, and those amino acids have a functional significance. And the way we find those is that we assume things that selection has acted strongly on for changes that have occurred probably have a functional impact. And so, there are people that have made their careers, the main 3 people that have developed these programs for finding these positions and alignments are Zhiheng Yang at University of College London, Simon Frost at Cambridge, and Sergey Pond at UCSD. And so they, especially Sergey and Simon, have come up with a suite of programs called Hi-Fi that have a whole set of different programs you can use for different types of analyses you're trying to do. Maybe you have more species represented or less, or maybe they're more divergent or less divergent. And so they've basically come up with a custom tailored test for each kind of scenario. And these are very valuable programs. And so, time and time again, virologists have demonstrated that these rapidly evolving positions in host genes seem to commonly dictate interactions with viruses. And, we assume that's because pressures from viruses drove them in the first place. So we took this Niemann-Pick gene from these species and we ran these programs. And like I said, they all have different strengths and weaknesses. So in the paper, we show this diagram of the receptor and we show all the different positions that were found to be rapidly evolving by these programs, not just sort of neutrally acquiring mutations, but really under pressure to change over and over again. And many of those actually fell right in this loop C that's on the endosomal side of the membrane where the virus contacts it, and which Kartik had previously shown was important for virus interaction. And, just amazingly, truly amazingly, that we ran 4 programs to find these rapidly evolving sites, and only one position in the protein out of how many ever amino acid positions Kartik said, a lot. Yeah. Only one showed up in all 4 programs. And so we made that mutation in the receptor, and sure enough, that completely accounted for the protection of the Edelon-Helvam cells against this virus. And so that was really just astonishing for us. Yeah.
So it's kind of funny how, science works. I didn't talk to Sarah for a while because, we're busy doing other things and they were off doing this. And in the meantime, Esther, one of my students, was doing something that she does very well, which is basically, thanks to Sarah, we had the sequences of the Edelon-Helvam NPC1, and we used the Rosetus aegyptiacus, which is just another one, just as a control. And she just systematically started swapping bits of these NPC1 genes in the background of the Edelon-Helvam to see if a piece of the Rosetus would actually complement, right? And she did this using a binding assay with just the loop C of NPC1 as a soluble form that I mentioned earlier. Mm-hmm. And she just swapped in these little pieces, and we'd gotten to a piece that was 3 amino acids. So you could take Edelon-Helvam and just put in 3 amino acids from Rosetus aegyptiacus, and you got a completely functional receptor as far as the binding interaction went. And, that's kind of when we heard from Sarah that, you were
doing this without...
Right. We're doing this sort of independently. We're doing the virology and Sarah's doing the genetics. And, when we're down to almost 3 or 4 residues, Sarah, tells us about this single residue 502. And I look at the piece that Esther's identified, and I'm like, wow, it's really that, it's in there.
That was your expression, wow, or something?
I was going to, I didn't know if we were FCC regulated or whatever, but...
anything goes on TWiEVO.
I swear a lot, so in lab, and I guess people are used to it. But so anyway, it turns out that, we sort of biochemically mapped that same residue that Sarah identified in her genetic analysis, and that was sort of an amazing moment where we really felt like we had something.
Really nice when these things converge on each other like that, the biochemistry with the evolution. Go ahead.
So one amino acid accounts for the relative resistance of these straw-colored bat cells to Ebola. All the Ebola viruses are just one?
So there's a gradient. Okay. There's another virus called Bundibugyo.
Right.
And it's a little bit like Ebola on the Edelon-Helvam cells. But then there is another virus called Sudan that's more divergent. And Sudan doesn't seem to really care. So it can infect these cells just fine. And Marburg is even more distant, and Marburg doesn't care. And there's this weird virus that was discovered in Spain a few years called Yovio, in these caves where these large bat die-offs were observed. And although they actually never isolated the virus as a replicating sort of particle, they were able to get almost a complete genome sequence. And this is a novel filovirus that's sort of equidistant from Ebola viruses and Marburg viruses from in an evolutionary and genetic sense. And that virus also, we know almost nothing about that virus in nature. That virus also doesn't seem to be affected by this one mutation, which by the way, from George Gao's structure, we can see where this mutation is. So it turns out that NPC1 sticks these 2 loops down into the receptor binding site in the cleaved form of the Ebola spike protein. And 502 is right there, is in one of those loops. Cool. And we've actually identified other genetic changes that have those kinds of effects, and including some in other kinds of bats. So we have some work now that we're doing with Sarah on Asian bats. And we're finding that some cells from some Asian bats are resistant to Marburg virus infection, even though Ebola can infect them. And it turns out that those differences map to the other loop that sticks into the receptor binding site. So, so I definitely think, we both think that we're really onto something here in terms of the evolutionary story.
So, Sarah, if you look at all the available NPC1 sequences, do you find this resistant residue anywhere else?
You do. In some other, one of the problems was we were able to get many more sequences to analyze from more species than we were able to get cell lines to analyze. Those are actually much more difficult to get. And so we haven't sort of tested all possible Niemann-Picks that have that residue. We do see it elsewhere. But I wanted to tell you, we didn't put this in the paper, but we also did an analysis of Niemann-Pick from primates, because sort of as a control, but also, it's been kind of postulated that, maybe primates could play a role, some primates as the reservoir of Ebola. So we just wanted to see what's going on there. And the protein is, amazingly conserved. Nobody's mentioned this, but this is your cholesterol transporter. So this is, obviously an important protein that's doing other things. And so it was a very different scenario in primates. I also wanted to say, a lot of people, when we think about zoonosis and the movement of viruses between species, I think most people tend to think about the immune system as being a major dictator of that, and MHC and antibody repertoires, and certainly restriction factors inside the cell, which the HIV field has done a really nice job of demonstrating act as strong determinants of species tropism for viral infection. But I think in recent years, it's really become clear that viruses interact with their hosts in 2 ways. They have to run away from all the immune detection that the host is trying to impose upon them, but they also have to run towards all of the host proteins that they need in order to do their life cycle and to make more copies of themselves. And certainly Niemann-Pick is an example of that kind of protein. This is not an immunity protein. This is an important protein doing its thing in the cell. And viruses, being the minimalists that they are, have to try to steal from the host all the things they need to replicate and make more copies. And, what we're finding is that these proteins, these some people call them helper proteins or dependency proteins or cofactors, these are just as hot of arms race battles as other things. And I like to give the analogy, every castle has a moat around it, and you defend yourself across the moat. Remember how castles have the little holes cut in them so that people could point their guns or their arrows or whatever they were using out the holes and fire across the moat against the enemy that's trying to come across. I really think that inside the cell, as the virus is coming into the cell, I think there are 3 main moats across which these evolutionary battles are playing out really intensely. And I think the cell membrane is one of them. Viruses have to get across that membrane. So this is a hot battleground for, evolutionary tit for tat evolution. So, and then I think the next barrier is the nuclear pore, because many viruses have to get into the nucleus in order to replicate. And that's a tightly regulated membrane the traffic across that membrane is tightly regulated. And one of my postdocs, Paul Rowley, has been working on this really hard. He just left to start his own lab at the University of Idaho, and I think he's going to have some pretty awesome stuff to put out on that soon. And then actually, we've been finding a fair number of arms races between viruses and DNA repair proteins in the nucleus, particularly DNA viruses that they come into the nucleus without telomeres. So they look like broken DNA, and the cell launches these massive assaults against them, and they have to do all kinds of things to protect themselves. So besides immunity proteins, I think we're going to see some really interesting things to come from those sort of other categories of genes.
Yeah, I totally agree, Sarah. And maybe even a few more moats there. And since we're sort of on this, battle footing with our metaphors. Maybe we could just take 1 minute to talk a little bit more about the idea of these arms races, because one of the things that I think a lot of our listeners might be thinking right now is, well, wait a minute, how could this really play out as sort of a back and forth arms race when you have these viruses with super high mutation rates, large populations, short generation times? How could a host even compete?
Yeah. So do you want me to take that one?
Yeah.
Yeah.
If you'd like.
Okay. Yeah. This is one of my favorite questions, and it took me a long time to wrap my head around that. It's studying evolutionary biology is kind of like philosophy, right? You have to kind of mull these things over for a long time, but I think I get it now. So there are two aspects of this, right? So you have an arms race between something that evolves very quickly, the virus, and something almost instantaneously we think of viruses evolving, and then hosts that have generation times of, mammalian hosts, years to decades. And so how does that work? How's there even a race to be had? So there are two important aspects. First, the mammalian immune system is so immense. It has a million guns pointed at the virus. And so the virus doesn't just have to evade one thing, it has to evade many things. And many evolutionary pathways are not going to be available to that virus because it puts it in the line of fire of a different gun, so to speak, because the immune system is just so vast. And so viruses are actually, while in the lab, they can mutate to all kinds of things. The number of those mutations that are actually viable in an organism are very, very small. So the pathways and the possibilities are very limited. The second thing is that you have to think about population genetics. Imagine that, we're humans, we get a lot of viruses. Imagine that, here's a perfect example. There are a group of humans that are resistant to HIV. There are people out there that have the CCR5 delta 32 mutation. It's a truncated form of the HIV co-receptor that doesn't express at the surface. And so these people, if they're homozygous, they're completely resistant to HIV. Well, has HIV just instantly evolved resistance to that and learned how to use a new co-receptor? No, because there are 7 billion more people to infect. There's no strong selective pressure on HIV to be able to infect those particular individuals. So, those mutations that, protect people from HIV will slowly rise in frequency as HIV tends to pick off susceptible people. And, maybe someday if CCR5 delta 32 people inherit the earth because the rest of us die from AIDS, now there will be strong selective pressure on that virus to utilize that other receptor. But that just illustrates the very slow dynamics by which these things play out.
But also you have to consider that humans take measures to prevent infection, which animals don't do. Bats don't do that, right?
But we also do things that allow us to get infected. And so I don't know, it's probably a wash. Sarah, since you brought up humans, I don't know. It's also really interesting, I think, with the Niemann-Pick story to think about humans because, there is this rare genetic disease called Niemann-Pick disease. And this happens when you inherit a different mutation usually from each of your parents, and you have essentially no functional NPC1. And this results in, essentially it's a horrible disease that is usually fatal in at least in the most severe cases. But I think the fact that there are all these, it seems that NPC1 can be mutated in such a way that it can still work, to do its normal cellular job of moving cholesterol around, while at the same time becoming at least somewhat resistant to Ebola. I don't know. It really has us thinking about the possibility that if we did, if humans have indeed been under sort of evolutionary pressure from Ebola, that maybe if we looked in the right place, we would find people that have polymorphisms in NPC1 that that confer resistance. In some cases it might even be that these are Niemann-Pick heterozygotes, because we know that if you take a mouse that's NPC1 heterozygote, so it's only got one functional copy of NPC1 as opposed to the two that normal wild type animals have. These mice are surprisingly resistant to Ebola. They get sick but they recover. So we think that for some reason we don't understand, the virus is being slowed down enough to let the immune system come in with its million big guns and blow the virus away. So we are sort of idly speculating that maybe there are maybe people with mutations in NPC1 or even heterozygotes in certain places where, humans might be under more in contact with Ebola, but, really have no data, and maybe it's not actually true. Maybe it's more like what Sarah said about the primates, which is that, there isn't really any kind of, since we're dead-end hosts, there isn't this process happening with humans. What do you think about that, Sarah?
Yeah, it's hard to envision. The problem is that when we talk about arms races, we're talking about, deep time. These things play out. So who knows what the filovirus landscape was a million years ago, and what humans faced at that time, or human-like creatures faced at that time. Who knows?
Yeah.
And I think, Kartik, it raises another interesting point, though, especially about the sort of heterozygote scenario. And that is that, what about expression levels? And so I was kind of, I couldn't help myself picking through your supplemental figures. And if you look at just the expression of NPC1 in the straw bats, at least for the Western blot that you showed of protein expression, it looked like it was lower. And so did you ever chase after that, the idea that if you modulate the levels of NPC1, in addition to the amino acid substitutions, that might be playing a role in some of this resistance?
Yeah, we definitely do see that in some situations where if you really overexpress a deficient mutant, you can get a little bit more infectivity. So, I think that is actually a real phenomenon that could be playing out in these heterozygotes, as you say. And we also wonder if, in fact, we don't really know this very well, if there are really big differences in the expression level of NPC1 in different tissue types in the body that could influence the susceptibility of those different tissues. This is not something this is something we wanted to do for a while, but haven't gotten around to it. I think the problem, of course, with the Eidolon Helvam cells is we're talking about a cell line. And so I think an important thing to point out is when we just express the receptor from that bat and look at the interaction with the virus, it's very, very weak. So I think it's weak enough that it's not zero, but it's very weak. And I think it's weak enough that, even if there was a lot of NPC1, we would still see a defect.
Yeah,
but
absolutely.
I think it's quite possible, I think that's something that we really do need to do, which is to, if we can get our hands on some more cells that, even tissues from real bats, I think that would be optimal to see if, what the levels are in different kinds of tissues, or even overall to see what role that might play. So that's kind of an aspect of, the analysis that I think is, we need to explore that isn't really done very much in this paper.
Mm-hmm. So the single amino acid change of 502, as far as you can tell, has no impact on cholesterol transport. Right?
Yeah, it appears to be completely fine.
So do you think this is a consequence of selection in this bat by an ancient Ebola virus infection?
Yeah, I think that's what we propose, which is that this mutation has arisen, as a consequence of an arms race with an NPC using filovirus.
So that means that the other bats that don't have this change have not been infected, or it hasn't been selected in them yet? Or can you
Sarah,
make any...
sorry. Yeah, do you want to jump in there, Sarah? Or?
Well, I just wanted, those are, so another way to look at this is, maybe, one thing some of our listeners might be thinking is, well, if the big mystery in this field is what is the reservoir, why do you care about this bat that's resistant? Isn't that ruled out? And so we've ruled out one, we have 977 more bat species to look at now. So, but that's not actually what Kartik and I have been, sort of the idea we've been tossing around is what if this bat was lucky enough to get this mutation that gave it some level of resistance? Well, we show in the final figure of the paper that a single mutation of in Ebola, in the glycoprotein, can restore the usage of the E. helvam receptor. So you really have to scratch your head and ask, why hasn't that happened? Why hasn't Ebola acquired that mutation and, exists in that form to infect these bats? Or, why would this be an effective resistance strategy at all for this bat if the virus only has to, get one mutation to escape it? And so one of the things we've thought about is that maybe this bat presents an ecological niche for the virus to exist in a slow burn form, which is maybe what it needs as a reservoir. Ebola is pretty frickin' deadly to many of the things that it infects. And so that's not like a great scenario for sustained maintenance in nature. And so maybe you need a reservoir species where you can exist at a slow burn. I'll tell you, if you look at some of the papers where people have isolated viral fragments from bats, that Ebola has never been isolated from bats, but a few genome fragments from Ebola have been isolated. And they mention in the paper that, and including this bat, I believe, was in one of these papers, they mentioned having to use serial rounds of PCR in order to get these viral fragments amplified, which would suggest that these viruses are somehow persisting at extremely low levels. Maybe that's why we've never found the reservoir. Maybe the reservoir is sustaining this virus at a very low level, and the genetic properties of this E. helvum bat paints a scenario where this could be true. Now, why would the virus not acquire the mutation that allows it to escape receptor control in this bat? It probably does, because viruses are mutating all the time. But I'm just going to speculate here that when that happens, those variants make these bats very sick, and the variants die out very quickly. And what's left is the slow burn variety that existed before. And that's how the reservoir maintains itself. This is all total speculation, but this is one way that you can think about how this might work.
I mean, there are if you look at the residue that mutates and allows better infection in the Eidolon helvum cells, that residue is not found in any other known isolate of Ebola virus, but it is present across the board in other viruses like Sudan virus, for example. So, again, if you want to continue this sort of, if you want to continue speculating along these lines, I think you could maybe think that mutation arose in Sudan virus as a response to a receptor mutation that reduced infection, just like Sarah's saying. And maybe in that other host, it's not as much of an issue. I mean, I think it's probably important to say that, there are many complicated things going on, and this is only one aspect of it. I mean, so for example, Paveshka's work shows that you can't really get Ebola virus to replicate very well in Rosetta aegyptiacus bats. And, Rosetta aegyptiacus cells are completely susceptible on the cellular level. So just because a cell is susceptible, doesn't mean that the host is also going to be susceptible. There are clearly other determinants of susceptibility in those bats that we haven't identified yet. So I think we're looking at one factor in a more complex sort of landscape. And what's true in one virus bat sort of situation, in one relationship, doesn't have to be true in another. I mean, you could have these different parallel paths happening just depending on the specifics of the virus and the host in question.
Yeah, and I mean, and as a testament to, the difference between what you find in the lab and what you find in nature, it wasn't actually, I remembered, it wasn't this bat that viral RNA fragments were isolated from, but an A. helvum bat has been found to be seropositive for Ebola. So, we're showing it's resistant in the lab, yet antibodies have been detected in bat. And an interesting thing about that paper where they found that is that they radio collared that bat that was that tested positive for Ebola antibodies, and it was still alive many, many months later. I think this was in Ghana. And so it doesn't appear that, bats really get that sick. The evidence is that bats don't get massive die-offs or anything from Ebola.
Although there is, these bats in Spain that died, may have died from Yovio virus. So that may not necessarily be true for, one mutation in the virus could make it highly virulent. So
this particular straw-colored bat, though, it's not completely resistant. So it does support a low level, and that may be what's going on here. And as Sarah said earlier, it could act as a transmission vector, but it doesn't die because it doesn't have enough virus in it. Right?
That's right. I mean, the other thing too, I think, is that it's also possible that the host is not the bat, but some parasite or something that lives on the bats, and the parasite's constantly biting the bats. Maybe it's injecting a little bit of virus each time. So the bats, develop an immune response.
Sure.
And so they become seropositive. And maybe those injections of virus are enough to kill the bat or make them really sick and throw off their breeding so that, over time, as Sarah says, in deep time, you can get these arms races, even if bats weren't sort of maintaining the, the virus as a reservoir. I don't know. It's just all rampant speculation, but it's fun to think about these different scenarios.
It sounds like you need a lot more field work to be done, because you need to look at lots of bats, especially these, and see what's going on.
I mean, this is like looking at a landscape painting through a straw.
Yeah.
I mean, we need a lot more stuff for sure.
Actually, Vincent, our peer reviewers agreed with you entirely. We were asked in peer review to find an Ebola-infected bat of this species, and also to experimentally infect E. helvum bats with our Ebola strain carrying the escape mutation. So,
My friend in Uganda could probably do it.
Yeah.
I mean, he puts mist nets out in his garden and he gets bats every night, supposedly. But I think it's still important, I feel like in the end why, this paper got published in eLife, one of the reasons was because this is really the first time anyone's identified a host gene, a host genetic factor that determines susceptibility in any kind of animal, really. So, I mean, admittedly, it's at the cellular level. We haven't proven that this carries forward to animals. But nevertheless, given this sort of huge amount of data that all kinds of different mammalian cells are susceptible, here we are showing this species-specific, resistance that's also very dependent on the virus. So I think that is even if we are looking at the landscape through a straw, I think that's sort of an important part of the story.
Yeah, I agree. And I'm really enjoying the conversation because I think what it illustrates is, some of the great points that both Kartik and Sara are bringing up here about kind of as you look more closely at or go toward the field or go toward the wild, you can start to just get a sense as you peel back the layers of the onion, how much complicated stuff is going on, how many variables. And yet at the same time, that feels like there's some tension there. But then using the biochemistry, the virology, and with the evolution, you end up still getting this insight despite all of that complexity. And so it's sort of this really nice back and forth, which, to me is really exciting. To peer reviewers, as Sarah's pointing out, cannot always be the most optimal situation, right?
Yeah, they're just trying to slow you down so they can publish first. That's the problem. Nels, don't go doing that onion skin stuff with me again. You know what movie that's from?
No, where are we going with that?
No, that's from Shrek.
I thought you were going to say Scarface or something.
Yeah, me too.
I have to plug eLife. It was I probably I'm biased because they published our paper, but it was such a nice experience. Just the way that it's so civilized. Usually you get reviews back and there's like 6 pages of like everything from this paper is complete crap, you should get another job, to you didn't put a comma over here, and it's all just a potpourri of like steaming reviewer exhalation. But in eLife, they actually have a conference call with the reviewers and the editor. They organize all of it. They like prioritize it, and they give you something that's digested, and they tell you exactly what they want you to do. And it's just it was such a civilized experience compared to some of the other places I've tried to publish before. So, this is probably going to be my only eLife paper ever, but
you don't say that. You never know. But also,
but I'm definitely going to send more things there.
They are open access also, so
that's also
our
really
readers,
nice
our listeners can check out the paper if they want, which is really nice. I think that's an important point. There's one last thing I wanted to touch on. You discussed in the paper some analysis you did on endogenous viral elements, filovirus-like related elements, which allows you to date, perhaps, when these viruses first entered bats. Is that your job? Was that your work there, Sara?
Yeah, I did recruit the help of Rob Gifford with this. He's really sort of the one of the main people in this field that's kind of been termed paleovirology. So it's very interesting. I'm not exactly sure who coined the phrase virus fossils, but as someone who originally worked in the HIV field, this was something that was very very obvious and understandable to those of us in that field. But HIV integrates into the host chromosomes as part of its life cycle. So as a result, there are 500,000 remnants of endogenous retroviruses in the human genome. They've left pieces of themselves behind for us to discover and try to understand their history. It turns out that every virus, it appears, has the potential to be captured into host chromosomal DNA at a very low level. Sometimes, this is mediated by line elements that, are genetic parasites in mammalian genomes, and they can capture RNA and reverse transcribe them and integrate them into genomes. But we're not exactly sure all the different mechanisms by which this can happen. But Rob Gifford and people in his field over the last decade have been cataloging all of these interesting virus fossils that are present in these mammalian genome sequences that we're now generating all the time in genomics. So we sequence mammalian genomes, and then inevitably what happens is that people run gene finding programs on these sequences so they can find all the genes in whatever bizarre species you've just sequenced. But then the next thing they do is start running programs like Repeat Masker to find basically viral type elements in these genomes. And you find a lot of things. You find things that are supposed to be there, like the lines and the endogenous retroviruses, but then occasionally you find really bizarre things. And so this is really wonderful because in this instance, there is a single insertion of a filovirus genome that looks like of a filovirus genome in the genomes of 3 related bat species. And the interesting thing is that the insertion is in the exact same genomic location in all 3 species. So what that tells you is that that insertion, that accidental capture of that viral genome happened before those 3 species speciated from one another. So there's really, in evolutionary biology, there's no harder evidence than that regarding the dating of something like, when a virus came to be or how old a particular virus family is. And so allows us to understand how long bats have been seeing these viruses in their environment, which is for a really long time.
Yeah, one of the cool aspects of this is that because the split, and so one of the really neat things that I didn't think about or anticipate was that a lot of the bats that have these filovirus endogenous elements, as well as some like this 502 mutation in NPC1, aren't actually African bats at all, but American bats. And so, no one's ever found a filovirus in either North or South America or Central America. But we're sort of thinking that, in deep time, maybe the North American bats were a hotbed for filoviruses. And, it's not clear where they went. Maybe they're still there. But it's just really cool to sort of see that signature in a genetic sense in these bats. And so Sarah's talking about this endogenous element. It actually predates a split between the big brown bat and the little brown bat or something like that. I'm trying to remember exactly. So it's like 22 million years, and we know pretty well due to work done by bat biologists when that split happened. So yeah, I'm really wondering if somebody will ever, if we'll ever find an American filovirus. We haven't yet.
Well, we can keep looking, right?
Right. Should keep looking.
Yeah, we should, for sure. And what you said at the beginning, you weren't sure why you did these experiments. So that is the way science works, right?
Yeah.
You just try something and see where it goes. That's why we have to be left alone
right.
to do that,
play.
right?
Yeah.
Don't tell us what to do. Let us figure it out. I mean, it sounds a little arrogant, but I don't mean it to be. It's just a point that smart scientists will come up with good things to do. Right?
Yeah, right.
And then mixing a few sandboxes together, it starts to amplify the fun.
Yeah,
definitely.
That's right. So are you okay with this, Nels? Should we move on?
Yeah, fantastic. This is a great conversation.
I think we ought to... there's one letter I think I'd like to read here, which these guys might like, actually.
Oh, yeah.
That's from Brian, who I think you know, right?
I do. Actually, I think Sarah may as well. Do you know Brian Wasick? He's in Colin Parrish's lab now, Sarah?
That name sounds familiar, but I can't place where I've met this person.
Okay. Well, he wrote us a letter, and it's here, Brian. Really excited by the new podcast, off to a great start, and I really love the tone and style of measured presentation you are both giving the subject. This is something Nels and I discussed briefly at ASV, if you remember, Nels.
I do. That
the field of evolution and this modern synthesis is very hot, and we see more of it within meetings traditionally covering microbiology, virology, or genetics. But there can be an over-eagerness to ascribe to every phenomena we observe a just-so adaptation story. No doubt there are some beautiful examples of adaptive evolution that capture something deeply inherent in us, rings some perfect chord regarding our view of life's grandeur. But so often things are more messy, more complicated, more drift, more hitchhiking, more non-adaptive processes, less sexy. It may be dour, but I often remind students and colleagues that the overwhelming story of life in this planet has been unmitigated failure and death. Well over 99%.
It's like sounds like it's writing R01s or something.
Now we're talking.
There is no progression, but it does make those little successes, ourselves included, all the more fascinating and important. A quick note from something that was said in the preamble to the topic paper discussions. Vincent noted that RNA recombination also occurs in nature and has been the source of variation in RNA viral populations. This can occur in homologous and non-homologous fashion. Nice examples include the historical diversity generation of alpha viruses between Old and New World species, Scott Weaver, and intraserotype recombinants in dengue. But as an example, Vincent noted vesicular stomatitis virus. Curious enough, negative strand viruses appear to undergo very little recombination. This is actually a big part of why myself and other researchers have used VSV as a good model for experimentally studying virus adaptation in the lab without that background. Eddie Holmes has had negative strand RNA virus evolution on his mind as a passion project and has been really digging for more sampling and working out the phylogenetics. His recent look in insects shows this virus genomic strategy is far more diverse than we thought. So how did this diversity come about with low recombination? Again, this goes back to your tone and something said on the show. How we teach biology needs to be assessed. Nothing is static. We don't know everything, but that's all the more reason why students should be engaged with that wonder and people should want to join this field. I like that. I think that's really good.
I agree. Thanks, Brian, for weighing in. And I do remember that conversation we had at ASV. And I think it's an important lesson to just sort of keep highlighting and hammering on the podcast, is that especially folks, myself included, who trained more in molecular biology and cell biology and kind of are coming to the ideas of evolution a little later in the game, is that it's sort of tempting, as Brian points out, to sort of see this grandeur and everything. He's quoting from Charles Darwin there, I believe, and to sort of put an adaptive explanation to every bit of variation we see. And that's sort of a dangerous spot to be in, because even the things that we sort of hold up in our paper from today included this one amino acid that can make the difference in this interaction between Ebolavirus and NPC1. There's tons of variation that Sara and Kartik sort of tested and discarded as not being important in that interaction. Maybe it is in another one, or maybe it's not at all.
Right.
And so that's always something to keep in mind. There's a great review that I think I'll add a link to it at the end of this episode, and should be sort of required TWiEVO reading. This is from Hopi Hoekstra a few years ago. And it's on this idea of seeing molecular spandrels. It draws from this architectural form, the spandrel, which is this sort of triangle in the arches of temples and whatever architecture. And we look at it, and there are these highly decorated objects, and people say, "Oh, wow, that spandrel is so amazing and the thing and super functional because it's sort of the point of the architecture." And in fact, it's just sort of this added-on feature that you have to have a triangle if you've built the arch, and it's just sort of post hoc decoration that sort of draws your eye but fools you into thinking it's important structurally when in fact it's not. And so, anyway, Hopi and others have drawn this analogy as sort of a cautionary tale. And so thank you, Brian, for that great note.
Yeah, but I would say that, you can construct stories about how mutations in certain genes came to be selected. But, the bottom line is the hard fact that this mutation matters for species tropism. And there's no concocting stories there. I mean, at least in cell culture, it matters. How it came to be might be somewhat of a story, but it does matter for species tropism. And there are some really practical things that can come out of that knowledge. Like, imagine that we did identify which bat was the reservoir for Ebola. Well, maybe we could take some simple public health measures to keep people away from those bats and stop these Ebola epidemics from happening. So working on things like this have very practical applications. I'll also note that things like what Kartik and I have described in these bats have a flip side that's very important to molecular biology, and that's that genes that create species barriers to viruses play into the choice of model organisms that you need in the lab to study a particular human virus. And so, we know that the same genes that have kept HIV and SIV from moving between primates are the very genes that keep us from being able to use macaques or other desirable non-human primates as model systems for HIV. The same genes that matter in nature also matter in the lab. And so, I think, one of my postdocs, Nick Meyerson, has been working really hard to show that this translates to choice of model organisms. So all I'm trying to say is, while it is really fun to tell and study evolutionary stories, I think there are really big practical things that come out the other end.
So I agree. Good points.
You guys, can we expect another collaboration paper from you two?
Definitely. Yeah.
Yeah.
Yeah. Well, we're
still working together.
We're definitely gonna have at least one more Ebola related, Ebola NPC1 related paper coming out, and then I think Sarah and I have been talking about more broad, sort of looking more broadly at other host virus interactions, not just with filoviruses, but other. I'd like to extend this story out, and I think look at other interactions that matter to the virus that are these so-called proviral or dependent interactions, or even maybe a restriction or sort of cell autonomous immune, innate immune interactions that could affect host range. And so we're still kind of throwing stuff around, but I'm hoping to do a bunch of stuff with Sarah, unless you're sick of me, Sarah.
No way.
It's just far enough away that doesn't matter.
Yeah, actually, Sarah's coming to visit Einstein. In mid-March, so that'll be fun.
Cool.
Yeah, that will be fun.
Well, by then you'll be past the snow.
Yeah, get rid of Snowzilla for me.
Yeah, you never know, right? So it could snow in March.
I want to tell you a brief story. It's kind of a similar story to what you guys have done here, but with polio. When I first came here, I was working on a strain of polio. There are 3 serotypes. I was working on a type 2 strain, which can infect regular non-transgenic mice. And we found that you could take 8 amino acids from the capsid of that virus and transfer it to one of the other polio strains that does not infect mice, and that will now infect mice, right? So we assume that it's a receptor recognition,
right?
But we don't know what the receptor in mice is for that type 2 virus, and we've been working on it, but now we have to throw away all those viruses because type 2 has been eradicated, and CDC said you got to destroy all your strains.
Wow, yeah. Oh, man.
So we're thinking, how could we just produce the protein on its own and see if we can pull out the receptor that way? You could probably do that, but then you could never test it in the virus, right?
Right.
Isn't that kind of weird?
It's all sad.
It's too bad, but I understand it's the right thing to do. Nels, you want to make a couple of picks here?
Yeah, so science picks of the week. I decided to go with sort of this annual festival that's going on now. This is the Sundance Film Festival up in Park City, Utah. And one of the reasons I thought I would bring it up is actually a movie I saw from it last year called Meru, given our theme here of snowzilla, blizzards, and tough weather. So Meru is a movie about climbing this peak, the Shark's Fin in the Himalayas, which was, I think there were 30 years of failed attempts and then 3 climbers.
That was a good movie.
Yeah, you saw it?
Yeah.
Kartik? Yeah. So it turns out one of the stars of Meru, and I think the producer, is this guy, Jimmy Chin. He and I actually overlapped in college. So I also went to a small college as an undergrad up in Minnesota, this place called Carleton, and Jimmy and I think overlapped for 3 years. And it's one of these scenarios where you see what someone has done with their life since you've sort of diverged from your shared experience, and you just feel sort of humbled and just sort of reminds you of, like, you think you might be doing well in your career or something like that. There's always someone who will ace you out. Right, Sarah? We used to talk about that from time to time about our...
Yeah, absolutely. I love the scene in that movie where Jimmy is, 90% up that mountain and he's smoking a cigarette. Did you notice that?
Yeah.
Who does that, right? Don't you have to have your lungs in top shape for that kind of stuff?
Exactly. Pretty tough. So anyway, some snow-related wilderness trekking there. I think it won one of the viewer awards last year at Sundance. Great movie. I'll put the link up there. It's worth checking out. And then just kind of a funny note for Sundance this year. is we have, Uber and Lyft, these car services in Salt Lake. And so for Sundance, Uber launched this chopper service. So you could take a helicopter from the airport up to Park City. That quickly got grounded. I guess they didn't have the right permits. And someone pointed out that actually you can drive up to Park City in about 30 minutes, and the chopper took about 45 minutes. And so that got nixed. How about you? So that's my pick of the week, the Sundance and sort of these related stories. How about you, Vincent? What do you have on tap?
I'll tell you what I got. But first I want to say, Nels, I think you're acing it.
Oh, thank you. There's always, in any profession, or any human endeavor, there's always someone who's you can point to and say, wow, they're really...
you're doing cool stuff. I mean, this is a movie and a lot of people see it, so you may be impressed by that. But in fact, you're doing very cool. We're all doing, except me, we're all doing very cool stuff. All right? So don't minimize it. It's all important stuff. So I have a pick. It's a podcast. So I am actually an advisor on the board of Quanta Magazine, which is published by the Simons Foundation. They're here in New York City, and they support a lot of good research. And they have this magazine, which is a very broadly addressing magazine. It's in physics and math and science, pretty good articles in lots of different fields. Their goal is to try and get articles on things that you wouldn't read in the popular press. So if they wouldn't write an article on Zika virus because everybody else is writing Zika virus articles, for example. So the articles are quite good, but I don't have a lot of time to sit down and read them. But they now have put out a podcast, so it's called the Quanta Podcast.
Nice.
And they just have people reading the articles. They have one woman actually with a nice voice, and she reads them. And I like that because that takes about a half hour to read each article, and that way I get to hear them. And they're not just in life sciences, but in other fields as well. So you should check that out. It's pretty cool. It's a nonprofit foundation, and they do support research. So,
yeah, thanks for the tip. I had seen some Quanta magazine articles that were, on the internet. Really great stuff. And so it's nice to know that you can do it in a few forums there.
So the Simons Foundation is endowed by Mr. Simons, who is basically a hedge fund guy, and he's a mathematician who's figured out algorithms to make a lot of money, and he's put a lot into this foundation, and he supports a lot of research, and he has set up this magazine. He's hired people from the New York Times and other writers to work on it, and it's really quite impressive. And the advisory board, by the way, in addition to myself, Hopi Hoekstra's on it. So I'm hoping to say, hey, Hopi, I'm on the board with you. We want to come on TWiEVO?
Yeah, great idea.
That sort of thing.
Now we're talking. I like it.
That's what I like to do. Well, this is TWiEVO. You can find it at iTunes, and you can also find it at microbe.tv/TWiEVO. And we love to get your questions and comments. You can send them to TWiEVO@microbe.tv. I would love to thank our guests for joining us today. Kartik Chandran, who is at Albert Einstein College of Medicine. Thank you, Kartik.
It's been fun. Thanks for having me.
And thanks for coming by.
Yeah, it's, haven't been here in a while.
Yeah, and I'm going to take your picture in front of the wall of polio, which is right behind you.
Nice.
You haven't seen it since I built it.
I have not.
We'll take your picture. And Sarah Sawyer is at the University of Colorado in Boulder. Thank you, Sarah.
Yeah, thanks for featuring our paper and having us on today. It was really fun.
Our pleasure. We appreciate that you went to a studio, but your sound is good, even though you didn't get much of it to work there. Okay. Is it very far from your lab?
No. And actually, I've got two of my colleagues here with me, Nick and Mariska. They're sitting in Nick's in-home studio. He has a band, so we're in his music studio in his house, and I've got 5 guitars on the wall here with me. And Nick made a comment earlier. He's like, yeah, this is a pretty different scenario than what this room is usually used for. So,
well, you should get him to fix it, and then you can come back and join us for another episode and talk about someone else's work.
I'd be happy to.
That'd be a lot of fun.
Yeah, and maybe an instrumental interlude there, or you could even play us out.
A jam.
Nels Elde is at cellvolution.org. You can also find him on Twitter as L Earlybird. Thank you, Nels.
Thank you, Vincent. Great to be here again.
And I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.